Hey everybody! Today we're going to be talking about the aerobic versus anaerobic testing that we're going to be doing in our class. And when we talk about aerobes versus anaerobes, an aerobic environment simply means that there's oxygen present, whereas an anaerobic environment is an environment that does not have oxygen or it lacks oxygen. And with that in mind, we basically have 5 different oxygen groups that we can place bacteria into based on their different needs for oxygen and the way that they utilize it. So we have what we call obligate aerobes. And obligate aerobes are organisms that require oxygen and they will die if oxygen is not available. The opposite of that would then be an obligate anaerobe. And obligate anaerobes are organisms that cannot use oxygen and they also die in the presence of oxygen. We have kind of a middle ground organism, what we call a facultative anaerobe. And facultative anaerobes are organisms that tend to prefer having oxygen present, but if oxygen is not available, they can switch to an anaerobic process and then basically survive in the absence of oxygen. Those are going to be the 3 standard groups of oxygen-based groups for bacteria that we have, but we do have 2 other a little bit more rare groups as well. We have what we call the microaerophiles, and microaerophiles are organisms that require oxygen but in lower than atmospheric concentrations. So they tend to live in areas, again, that are exposed to oxygen, but it's a bit less than what we would see naturally in the atmosphere. And then our last group are what we call the aerotolerant anaerobes. And aerotolerant anaerobes are organisms that, again, are anaerobic in nature, so they cannot use oxygen, but they're also not killed by oxygen. So again, those are the 5 basic groups that we have. And when it comes to testing for different kinds of aerobes and anaerobes, really one of the biggest limitations that you're going to have when doing that testing is going to be funds. And kind of if you're looking at this in like a research perspective, you might be able to use something like this, these fancy anaerobe hoods with the fancy arms to stick into them so everything can be inside of this anaerobic environment. But really for our class, again, we don't get to play with anything quite that fancy, but we do have 2 different mechanisms that we can use in order to basically test for these different groups. So we have what we call an auger deep method, where we use auger deeps, and then we have our slant and jar method that we're going to be talking about. So really, depending on what materials that we have, we will either be doing a auger deep method or a slant and jar method. Just again kind of depends on our availability. But kind of with that in mind, let's start out talking about the auger deep method, the kind of the materials that you need, and then how it works out. So in order to perform the auger deep method, you're going to need some standard items. Again, you're going to need your Bunsen burner and striker in order to do your transfers aseptically. You're going to need a loop in order to do the transfer. You're going to need your sample, again, that you're trying to test to see which category it falls into. And this sample can be any type of sample, whether it's broth, plate, or slant, doesn't really matter. And then you are going to need some sort of setup in order to basically boil your auger deeps. So you will need a hot plate and a beaker so that you can boil the water and you can put the auger deeps inside of them, and then we can boil them. And boiling the auger deeps are going to serve 2 basic purposes. It's going to, 1, solidify the media. Again, this just has auger inside of it, so it is a solid medium. So we have to basically, melt the media, it's going to melt the media. That's the first part to it. It's going to melt the media or liquefy it. And by doing that, again, we can actually inoculate that media itself. And then the other part to it, the second part to it, is that it drives out all the dissolved oxygen. So it basically takes out all the oxygen inside of there and creates basically an anaerobic environment. And once you've done that, kind of the next thing you also have to have is actually a water bath that's set to about 45 degrees Celsius. And it has to be 45 degrees Celsius because that means that it is warm enough that your auger deep does not re-solidify. Normally our auger solidifies at about a temperature of 42 degrees Celsius. So we keep it just above that solidifying temperature, but at the same time, it's also really not hot enough that when we inoculate it with our bacteria, then the bacteria die right away. So it kind of has a little bit of prep work to the way that you set it up, But once you basically have your boiled agar deeps, they've been sitting in the water bath for a little bit to cool to that 45 degrees Celsius, the procedure is pretty straightforward. You basically just inoculate them with your loop, again, aseptically inoculate them with your sample, and then at that point you wait for them to grow. There is one thing the students tend to do though sometimes that can kind of mess up this experiment, and of course I forgot to grab a loop, but here we go. When it comes to the agar deeps and the way that they're set up, they are big enough that if you were again to flame your loop, you get your sample and you put this all the way down inside of the agar deep, you can kind of see how that the base of it does get down into the medium. And if you do that, since we're not flaming this part, it is going to have quite a few bacteria on it, and because of that you'll contaminate it with things that are not your sample. So make sure again then when you go through and you do your inoculation, you only go down into the media again about 80% of the way, and in that way you're not actually getting the base of your loop inside of there, you're only getting the loop part itself inside of there. But after that, again, once you've aseptically inoculated your boiled agar deep that's been sitting at that 45 degrees, you just leave it out at the table, and over the course of about 10 to 15 minutes it will basically solidify. And as it's solidifying, it actually creates an oxygen gradient inside of the medium. So there'll be no oxygen at the bottom and there'll be a lot of oxygen at the top. And because of that, what ends up happening is that we can actually test for all 5 of our different oxygen groups using this technique. So what ends up happening again is that if your organism that you put in there is an obligate aerobe, it'll only grow at the top. If your organism is an obligate anaerobe, it will only grow at the bottom. If your organism is a facultative anaerobe, it will grow quite a bit at the top, and then you'll see scattered colonies all throughout the tube. If it is your microaerophile, again, it will need oxygen, but in lower concentrations. So it grows about 20 to 30% down inside of your tube. And then lastly, again, if we have our aerotolerant anaerobe, it's just going to be scattered growth all throughout the tube. So this is a nice sort of easy way for us to basically go through and look at, with the different items again, or the different samples that we have, which of the 5 categories do they actually fall under again, just by inoculating our agar deeps, letting them solidify, and then waiting our 48 to a week or so in terms of that incubation period, and then just scoring them based on where's that growth sitting. So that's our agar deep method that we can use. And again, if we have the materials to do that one, we might do that one. If we have the materials to do the other one, then we'll do the other one. So let's talk about our slant and jar setup that we have. And basically, in terms of the slant and jar setup, the materials that you're going to need, again, you're going to need your standard items, Bunsen burner and striker, so that you can do it in aseptic transfer. You're going to need the loop in order to do your transfer, and you're going to need your sample. And again, sample can be broth plate, slant, doesn't really matter. Now, what you're also going to need is that you're going to need a slant, basically 1 slant per sample that you want to test. And then you're going to need a plate, again, and if you're doing multiple samples, you can put up to 4 generally on one of these plates here. But you will need a slant and a plate, and then you're also going to need our anaerobe jar. And generally, when it comes to the anaerobe jar, you will also need a candle and something to light the candle with, so just a lighter. But basically, the way that this techniques work out is that our anaerobe jar is basically a fancy mason jar that we pay $1,000 for because science. But the way that it works is that we would put our sample, basically, again, your basic procedure is aseptically put your sample on the plate, put it inside of the jar, light the candle, close the lid, and then the candle will basically burn off all the oxygen. And now your jar, or inside of the jar again, is going to be an anaerobic environment. However many samples again that you have, you'll inoculate those on the slants, and your slants are now aerobic because they're exposed to the air. So what you've done is that you've effectively grown your organism in an anaerobic environment and an aerobic environment. And what we end up looking at here then is, let's say again that I have 3 organisms that I want to test. So I put organism 1, 2, and 3 on my slants, and I put organism 1, 2, and 3 on my plate. And after my incubation period, again, my 48 hours, a week or so, depending on how we're doing it, I come back and I'm trying to score them. And let's say that organism number 1 only grew on my slant, but not on my plate. That meant that it could only grow in an aerobic environment, and because of that, it has to be an obligate aerobe. Organism number 2, let's say that it could only grow on my plate. So it could only grow in an anaerobic environment, and because of that, it's an obligate anaerobe. And then lastly, our organism number 3 could grow in both our slant and on our plate. So because of that, it grew both anaerobically and aerobically, and that makes it a facultative anaerobe. So the limitation that we have with this procedure right here is that it basically can only tell us those 3 main groups, again, obligate aerobes, obligate anaerobes, and facultative anaerobes. Now, when it comes to our class, though, that's not really that big of a deal because microaerophiles and aerotolerant anaerobes are pretty rare. We're not really going to be working with them. We're only going to be working with the 3 standard groups. So either one of the techniques work perfectly fine for what we're trying to do for our class. But I think really with that, I don't have anything else that I need to introduce on our aerobic versus anaerobic testing that we're going to be doing in class.